Welcome, everybody. We're going to get started in several minutes. We'll give some folks some time to join because we know how busy everybody is and what it takes to go from one meeting to this or to the next. But we do have a very action-packed, live, long event today. And it will be recorded, and I'll repeat that a couple times so people know. So if you're just joining and you hear this, you've got several minutes to go get yourself something to drink. You're not going to miss anything. Or get comfortable. Put an exclamation point on that last sentence in your email. All right, let's get started. So today we have a very special guest, Simulation Plus. And this is a webinar series. This is the first of 4 webinars that we'll be doing together. Today's title is Accelerating Small Molecule GLP-1 Agonist Optimization. AI-driven design meets mechanistic modeling for a first-to-invent advantage. So let's just quickly get into the agenda. So we'll talk about rules of engagement, the intro to Simulations Plus. There'll be a preamble and then the presentation, and then we'll go into a Q&A session with Scott and Jeremy. So really important, put your questions or comments in the chat or the Q&A box. It doesn't matter which one. The Q&A box is private. Nobody else sees it. If you put it in the chat box, people get to see your name. Some people are sensitive about that. We don't care. We'll address all questions. This is really important. This is going to be recorded. You might have to leave. This is actually going to take the full hour, maybe 10, 15 extra minutes. So it could be an hour and 15-minute webinar today. So be prepared for that. And it is recorded, will go on demand. So if you have to leave your questions, you can come back and get the answers to them, or reach out to the folks at Simulations Plus to take it further. So just be forewarned. The last thing is Jeremy is in New Zealand and he's tired. And so what we did is we pre-recorded his talk so that you're not seeing a tired Jeremy, and it's very clear and concise. And then, but he'll be here for Q&A. So just so everybody knows, we're not trying to pull the wool over anyone's eyes. So who is Jeremy? So Jeremy is the principal scientist of AIDD applications. Jeremy Jones has his PhD and he's a cheminformatics expert. He joined Simulation Plus in November of 22 as a principal scientist focused on early stage drug development and AIDD. Prior to joining the company, Jeremy was the director of oncology at Terray Therapeutics and led his own consulting company. So Jeremy's been around the block. He's probably seen all or most of it. Before that, he was a professor at City of Hope Comprehensive Cancer Center for nearly a decade, where his lab focused on drug development and translational research in urologic oncology. He received his PhD in microbiology and immunology from Stanford University and completed his postdoc fellowship at the University of California, San Francisco. He now resides on the North Shore of Auckland, New Zealand, where he enjoys time at the beach with his family. Impressive career, must say. Scott Seiler has his PhD, and he is the Chief Science Officer for Quantitative Systems Pharmacology. He is not only that at Simulations Plus, but he leads QSP and QST efforts, including the development of NAFLD Sim and IPF Sim, used to assess over 35 NASH and IPF compounds. He's also contributed to DILI Sim, a leading QST model for evaluating drug-induced liver injury in over 80 compounds. Previously, Scott spent 12 years at Entelos, leading metabolism modeling projects and evaluating treatment for type 2 diabetes. He holds a PhD in nutrition from UC Berkeley and a BA in biochemistry and molecular biology from UC Santa Cruz. So another highly accomplished person who's now helping bring scientific software to in silico first and very efficient approaches to solving some of our toughest problems. So with that, I'd like to hand it over to Scott to give the preamble.
Right. Thank you very much, John. Let's see, I need to share my slides. And so here we go. All right. So I'm really happy to have the opportunity to introduce this audience to our series of webinars, this being the first, of course, on how Simulations Plus and our various technologies can help biotech companies accelerate and increase efficiencies in drug development. Given how substantial the interest is currently in obesity as a therapeutic area, as a disease, we've focused our webinar series on this disease area in order to kind of serve as not only an example, but also a poignant one at that. For me personally, as John mentioned, I have a background as a nutritional biochemist, and I've been in this field, obesity and otherwise, metabolic syndrome field, for over 30 years. And so for me, this is something of a passion project, but also I think it's really important that everybody understands some of the key aspects about obesity as a disease and the drug development efforts that are now supporting treatments for patients with obesity. So weight loss really impacts myriad related conditions for patients with obesity. As the figure in the Kunti et al. Citation shows, it really nicely describes that weight loss can improve hazard ratios for sleep apnea, type 2 diabetes, dyslipidemia, PCOS, and many more maladies. Again, we often refer to this as the metabolic syndrome. They are intertwined, and in sort of a spoken hub concept, the hub really being increased adipose tissue that comes along with obesity. As such, treating the obesity, treating patients with obesity to encourage that weight loss really has impact beyond just the body weight itself. Really exciting because for years we had this supposition that in fact that would be the case. And here we are now at the precipice where we really have the ability to see it in real time and the precipice of even more. So with the relatively recent entry of liraglutide, semaglutide, tirzepatide has finally proven, or rather provided effective treatments for patients with obesity. Again, this is a welcome development. I mean, you can see as this nice inside scientific visual provides that we went decades without success in this area, and now we do. So again, super exciting with a great potential for impact, both for patients and on the business side, of course. Many of these and those initial triumvirate of medicines that are now available are really the backbone of them for all of them is the GLP-1 receptor agonists. And that comes with effective weight loss, but also some GI adverse events, nausea being perhaps the most prevalent. And so we're balancing those, perpetually. How do we get the best in efficacy with the least amount of adverse events? And one of the ways that we're pursuing that is by, as Melson et al. Describes in this figure, expanding the field of vision to other potential targets that also impact food intake and reducing caloric intake, such as amylin, such as peptide YY. The GIP and GLP intertwining relationship is a really an interesting one. And then glucagon that seems to as well move into another category, which is compounds and treatments and targets that address increasing energy expenditure. So a nice complement potentially to the food intake effects. So really we have all these possibilities in front of us. Many of, in the biotech and just across industry are pursuing these and really trying to understand, is this the right one? And how is this going to compete? Should we combine X with Y? All of these questions are kind of circling around. And frankly, one of the best ways to understand and then really focus the efforts in a very efficient way is by applying mathematical modeling. Again, Simulations Plus has a variety of modalities across this spectrum. We're now able to apply that expertise across that spectrum of mathematical modeling, as well as our expertise in the disease area, in this case being obesity, to really help support drug development across the way, across the spectrum, and enhancing patients and as well increasing business opportunities. So at this point, I step away. I pass the ball to my colleague Jeremy Jones, who's been properly introduced by John. And thank you for your time.
All right, awesome. Now, share the recording. Just to find what happened to it. It's 2 seconds. All right. It has disappeared on me, so I'm just gonna, there we go. I might have screwed that up. I apologize, everyone. Just try this again. Share sound. Not hearing sound.
So let's dive right in here.
There we go.
Talking about small molecule GLP-1 agonist optimization. So over the next, 40 minutes or so, I hope to highlight some of the capabilities of our sort of early drug discovery platform. And I'll do so through trying to answer 2 questions, 2 scenarios which you may be facing now, or it will at least show you might have related issues that you're looking into, that some of the things I show here will certainly apply to. So the first thing we'll talk about is what happens if you're thinking about in-licensing a novel chemical series, and what kind of tools could you use to start to do your evaluation to make sure that this series has the best chance of clinical success. And the second one we'll get to afterwards is, series looks pretty good, but it could use some further optimization. We're talking GLP-1 receptor agonist. Let's say we want to try to improve the penetration through the blood-brain barrier. So the first one, we're going to assume that a company's coming with us. They have you know, some limited SAR, some compounds that look very efficacious in cell-based models. So you have some in vitro efficacy data, and hopefully they have some in vitro ADMET data, and maybe even some limited PK at the very least. And I'll show you how we can use ADMET predictor to predict any parameters that we don't know, that haven't been measured. And then I'll show you our high throughput PK, our physiologically based PK module within ADMET predictor. And if you're familiar with GastroPlus, it's a very similar module, or very similar calculations to GastroPlus, just a lot faster. And how we can use that to quickly simulate PK in both animals and humans. Yeah, everything from oral bioavailability, systemic exposure, tissue partitioning, and a dose necessary to reach an effective concentration in your target tissue, things like that. So, without further ado, let's look at a hypothetical series of small molecule GLP-1 receptor agonists with ADMET predictor. So give me 1 second to switch over. Actually, I should, if you're not familiar with the ADMET predictor platform, let me start by showing how easy it is to import some data. And so you can import data or open data from several different files. It can be a structure file like STF or mol, or you can see all these different types of data here, even just a text file. So if you have, just the smiles, the smile strings of these compounds, it's as easy as opening this up. You can see that the software automatically recognizes a smiles field and an identifier field. And you can see, you have these different things load all the compound attributes. And in this file, my hypothetical chemical series, I'm going to make these rows a little bit bigger so we can see these molecules better. This is actually all of the GLP-1 agonist data that I've collected. In the second part of the talk, I'll show you how I use this to build a quantitative structure activity relationship model as a way to maintain activity as we're trying to optimize the blood-brain barrier. But very flexible platform, very easy to import and open data. And so here's the series that I was talking about. What you can see is this series of compounds, and we have the EC50 activity that comes from a cell-based assay. This is a cyclic AMP accumulation assay that's very standard in the field. And you can see these are really nice and potent molecules. In this series we're looking at, we have 10 molecules, 9 plus the parental molecule here. And so, a couple of the things we can do. Let's see. And again, I sort of pre-populated this table here. Let's say some of these, we have the in vitro data, but we also have some measured in vitro ADME data. We have a measured RBP, blood to plasma ratio, fraction unbound in plasma. We've measured the intrinsic clearance in human liver microsomes, and we've measured the thermodynamic solubility, real solubility, as it were, as well as the log P. So some of the most important elements for ADME endpoints for trying to understand how drug-like and developable a compound is, right? And for instance, we have these values for most of the compounds, but not all. So one of the first things we do here is, this is just the input we have. We are going to, well, you know what? Before we even do that, I want to show you a couple other tools. See, we have some compounds that come in. We want to make sure that they're all in the right form. So we always do some quick cleaning. We have the ability to remove the smallest fragments, to neutralize, to standardize the functional groups. And we have a really good tautomer standardization, which I've already done for this, to make sure it's in the most likely tautomeric form at a particular pH. And in all the default settings here are pH 7.4. All right, you can also go through and find if there's any duplicates and a lot of functionality here. But let's go back to what I was going to say. Let's calculate all the ADMET properties. And so when I hit this, you can see it comes up. And depending on what modules you have in ADMET Predictor in this platform, basically you're always going to have the physiochemical. We also have ones where we predict transporters and the metabolism as well as toxicity. We have a new DILI sim module here to predict some drug-induced liver toxicities. And if, as I'll show you in a little bit, some user models that we can create on our own, we can then import those and predict using those models. I also want to point out that Simulations Plus has spent a lot of time creating descriptors. And this becomes really important when you're trying to build models. And there's several studies out there. J&J actually just published a really nice one in the last year or so, showing how much value the ADMET Predictor descriptors add to model building. So we have some proprietary descriptors that really help. Again, just something you might not think about, with the name ADMET Predictor. Yeah, we've spent a lot of time on this. So let's see, these 10 molecules, this will go incredibly fast. You can also change the pH and do a bunch of other stuff, but let's stay standard here. So we're going to calculate all of these properties. You saw how fast that was. I mean, 10,000 molecules would go in a couple seconds too. It's just a very fast. And so we can look at the phys chem stuff, the different models we have, the different permeability models are solubility models. We have, many models, and depending on exactly what you're interested in, you can limit this. One really nice way to do this is we have this customization tab. So if you just want, we don't need all these yet. Let's remove these. We're eventually going to look at the blood-brain barrier. So let's do our BBB filter, blood-brain barrier filter. Let's do, we also have a log BB model. So the two different types of models in general, classification, it's yes or no, high or low, things like that. Also regression models that are continuous models, right? And so these are two models we have for the whether or not we predict a molecule will cross the blood-brain barrier. This filter is yes or no. The log BB is a regression model saying how much do we predict will cross. All right, so if we want to just really focus on a couple of these most important ones, this is a nice, easy way to do it. And we can look and see, many of these are predicted to have low blood-brain barrier penetration. We also give you sort of a qualification of how sure we are of those predictions based on the chemical space and how well our model responded. So all of these are predicted with high confidence to have really poor blood-brain barrier penetration. What else do I want to show you? Let me go back to all, because just thinking about these models and how confident we are in various models. I will show you. Let's find an example here. I want to go back to let's try the phys chem. And you might see some of these values flagged in red, right? And so what this means is we think that this specific chemical is out of the applicability domain of our particular model. So our models are built on typically hundreds to thousands. Our pKa model is the industry standard. It's built on more than 70,000 pKas from, we were lucky enough to have Roche and Bayer and Genentech all give us all of their pKa data. And well, mostly because they know our model works better than anything they can build in-house. And so, our models vary between 70,000 data points and several hundred in the case of the smallest ones. But all of these models will have an applicability domain, and it's basically defined by the different descriptors, how far out of range a particular chemical is from the different descriptors. And for instance, this molecule, we flash up in red that this, for whatever reason, is outside the applicability domain. So we are not as sure about this prediction. This just happens to be our fraction unbounded human hepatocytes. So actually a newer model that we built, which, you know, if you're working on hepatocyte clearance data, often you want to know how much of your drug is bound to those hepatocytes and correct for that. Just a random example. Okay, so there's a lot of other tools here to help you prioritize and figure out what is good or, you know, or bad. What are the deficits? One of the things I like to look at really quickly are our risks models. So let me scroll back. So we build up a couple different risk models, and this is sort of the overall. So this is, you know, is it a toxicity risk? Is it a metabolism risk? Is it an ADMET risk? And so, for instance, here you get our ADMET risk score. And so our ADMET risk is built on 22 different properties of our property models. And so you can get a range of 0, meaning that we're not predicting any ADMET issues, to 20 to predicting a lot. And, you know, the ADMET code here will tell you what we think those issues are. So if you look at like the World Drug Index compounds, the average, you know, it's a really nice bell curve distribution. And the average ADMET risk score is between 3 and 4. So, you know, these are drugs that have made its way through, don't have a lot of issues. So the ADMET risk score performs really well in this case. And you can look and see exactly where it's failing, right? And so these pop-up windows will tell you what all these different codes are. So this one is failing size. Yes, we know these molecules are big. It's predicted to have some toxicity in rat and mice. It's predicted to have a CYP3A4 liability and some clearance liability, right? The next one down tends to actually hit some, our PF model is our permeability model, right? All of these are actually predicted to have some 3A4 liabilities. Again, something we can optimize out with this. So these are, you know, sort of the in vitro ADMET properties, but what I think is a real differentiating advantage of this platform is not only can we look at the in vitro ADMET, we have really accurate in vivo predictions as well. So we bring in the ACAP model from GastroPlus and simplify it only a little bit. And this is the model that basically looks at the disposition of drugs as it goes through 8 different compartments of the gastrointestinal tract, accounting for the different pHs in each of those, as well as a central compartment in the liver. So you're doing pretty much full mechanistic PBPK modeling, but really fast. We actually worked with Roche a couple of years ago to test how this worked, and they had a really nice publication showing, you know, basically identical or nearly identical results to full GastroPlus PBPK modeling versus this high throughput version. And so all we have to do here is, I'll just go back so we can see our molecules again. The way to calculate all of these properties, the quickest is to hit this button. And you can see here, this is ADMET Predictor version 12. 13 is coming out soon. In 12, we have 3 species, rat, human, and mouse. In 13, we've added dog and monkey as well. We also have 3 different dosage forms, you know, an IR immediate release tablet, which, you know, you would maybe want to try to develop for humans. Let's think about maybe looking at a preclinical animal like a rat. You could do an IV bolus, or you can do what we call the immediate release solution. This is like an oral gavage solution that you would, you know, be feeding to rats. I'm trying to think of the parental molecule here. I know I have some data. I think at oh, actually, you know, when I just did this right before I started recording. We have data, yeah, it's at 25 mgs. And so I just ran this and I wanted to show you how well it worked. Let me go back. All right, so our dose is 25 mgs. in rats, and it's an oral solution. We'll just do 1 dose for now. A lot of these things that you can incorporate. First and foremost, if you have clearance data, we have the ability to do liver microsome, hepatocyte, or systemic clearance. Instead of using our model, this is our intrinsic clearance in rat liver microsomes. If you had a column such as, it's not going to pop, oh yeah, measured data, right? And you wanted to use your measured data instead of just our predictions, you can substitute that in here. And if you only have measured data for part of them, you can do the preferred values, the measured, and then the fallback to our predictive model, right? And so here you can see the other parameters that go into this high throughput PK simulation. The log P, solubility factor, this is our permeability model, fraction unbound in plasma, the blood to plasma ratio, and then different choices for volume of distribution, first pass extraction, and renal term here, all of which customizable. You can substitute in your own. We'll go with the defaults, fully in silico predictions at this point. And we'll run it for all 10 of these compounds. Let's see, hopefully this works. And so you saw how fast that was for 10 compounds. Even if you have 10,000 compounds, it's only going to take a couple seconds. The guys who wrote this code are geniuses, far beyond anything I could do. But yeah, they were able to make these really fast. And so what you see here now is all the different measures we have. So percent fraction absorbed, percent fraction bioavailable, oral bioavailability. And this is at a specific dose, right? So many compounds are dose dependent, and so it's really important to incorporate dose and route into these simulations. We predict the minimum concentration, maximum concentration, the Tmax, the AUC, whether to the specific time or calculated out to infinity. And you can see again these hover over boxes give you the details and the units and all that stuff. So you have the clearance, plasma clearance, the half-life, and then you can get the renal clearance term. You also get all of the KP values, so the partitioning values that come from the Gastro Plus calculations as well. And so, if you're looking for a drug that partitions well to the brain, actually this one is pretty good, has a nice brain to plasma partition ratio, but that could be for any of these tissues. And I'll show you in the next part of this talk how we're going to optimize for that as well. What else do I want to show you here? Oh, I know what I did. I did want to show you real quick. The data, it doesn't always work out this well, I promise. But the data that I had found for the parental compound here is the half-life, the oral availability, and the Cmax. This is what was measured. I pulled this out of Campbell, obviously. The half-life measured was 2.37 hours. We predicted just under 5. The oral availability at this dose was 39%. We predicted 30%. And the Cmax, you can see we were pretty close there as well. Certainly within twofold for all of these. Again, it's not always going to work out that well. I have a publication that I'm putting together right now that we put together a huge database of PK endpoints for all 5 of the species, and we did a bunch of different metrics to see how well our predictions did. Maybe the main criteria being the percent that fell within twofold, and we do better in some, worse in others, but our predictions are pretty accurate, and happy to share that data with you as well at some point. In any event, we did pretty good here, and that's nice to see that our HTPK, the high throughput PK simulations, are at least pretty close for the parental compound in this series. So that gives us some more confidence going forward that these simulations for related compounds are going to be pretty accurate as well. I will just show you maybe one other thing real quick before we go to the second part. Some of the other tools we have with HTPK. This is the estimated dose needed to reach the target concentration. So what did we have? We had the EC50 in nanomolars. So we can actually just put that there, micromolar, All right, should have done this beforehand. I have it nanomolar and we only have micromolar here. You can do total and unbound. You can target the C average. You can target the C min, C max. We'll do the C average here. I'm going to go back and you know what, I'm just going to put this in. So in micromolar, give me 1 second. Let's look and see user input. Let's go for 1 nanomolar. Let's say we want to hit 1 nanomolar, and we'll do all the compounds just to see what happens. All right, let's go for human this time, and what might be an IR tablet. Let's see. Let's jump way ahead of ourselves and say, hey, are any of these drugs going to be good orally in tablet form to reach our target concentration of 1 nanomolar? So that's 0.001 micromolar. The average, let's make it even harder on ourselves. Let's do the unbound. You gotta, the way our algorithm works is you need to put an initial dose guess, and I haven't run this, so I have no idea if it's gonna work. We'll see. If it doesn't, it will tell us that we're over and we need to try a different initial dosage guess. The other things we could do here are change the dosing interval if we, if we know the half-life. Let's go back 1 more second. Let's look at the half-life predictions here. So you can see how you would work through this. So our half-life prediction, for this one, it's actually 22 hours in humans. So the rest of these are 3. Let's go with, we'll do a dose, twice daily dosing. How does that sound? We'll try. All right. So back to human, IR tablet, target concentration, 0.001 micromolar, C average, unbound. Advanced, we're going to go dosing interval of 12 hours, so twice daily. Okay. I have no idea if this will work. Let's see. All right, so we didn't get the red flag, so we're not out of range. So the optimal dose to reach a plasma concentration, we can change this around too, this is customizable. To reach a plasma concentration unbound at 1 micromolar would be 0.638 mgs for this compound. Yeah, so there's, you know, if you wanted to do this for figuring out what the starting dose might be for a compound in a mouse or rat model, this is a great way to start it as well. You can also see how many doses we predict it will take to get to a steady state. And you know, the other thing we can do here is a full. We'll look at we'll do the same thing for this. Let's pick a random compound. We'll try this one. We can predict the full CP time curve as well. So we'll do the same thing. Human IR tablet doses. Let's do optimal dose to reach that concentration. Dosing intervals. We'll see what it looks like over 2 weeks. Advanced again. Let's do our dosing interval of 12 hours. And we'll hit run and see what the CP time curve might look like. So you can, actually not too bad. We reach steady state pretty quickly. I don't like pretty big variability. We're going from, actually that's not too bad at all. The C min, we're hitting 10 nanomolar to 40 nanomolar, maybe a little bit more than we need. You could consider scaling that back. All right, I've rambled on enough. Let's stop here and switch to the other part that I want to talk about. Okay, sorry for the slightly abrupt transition there. Back to this slide. That's the benefit, I guess, of recording this ahead of time. I had to stop and grab a glass of water. In any event, we talked about some of the tools that we can use to really dive deep on compounds and understand, not only their in vitro properties, but really give pretty accurate simulations of what they might look like in vivo in animal models or in human models as well. Just as a first glance at looking at it. But as we noted, and what I noticed as I was starting to put together these slides was that in this data was that a lot of these compounds had really poor predicted blood-brain barrier penetration. And so we have a set of tools that can really help optimize that. The first being our AI-driven drug design platform, AIDD. And what this is really a compound generation engine that uses Smerx-based transforms. And again, we worked closely with pharmaceutical companies to make sure these were, really relevant and chemically, consistent Smerx-based transforms. And that's coupled with a powerful multi-parameter optimization algorithm. And so you can do things like simultaneously optimize many properties like potency, ADMET properties. As you saw, we can bring in, we have the fast calculations of systemic PK, and in this case, brain distribution as well. After we design these molecules, we have several tools that can help you prioritize which molecules you want to send out for synthesis and testing. And, it doesn't have to be molecules you design with AIDD. It can be molecules, the compounds that came from a high throughput screen that you did. And you want to know, how do we prioritize for which subset are we going to send out for synthesis and testing, right? And yeah, so we'll show you some of those tools as well. Strategy here is, our goal here is, what I'm going to try to show you is how we can improve the brain penetration of these molecules while maintaining potency and good predicted PK. We're going to use AIDD to design some optimal derivatives. I think it's really important to mention, and I know, there's always this struggle for medicinal chemists when you're trying to optimize one parameter and not lose another parameter, right? And of course, you look at these molecules, they're really potent, right? Great efficacy, and we don't want to lose that. And so when you're optimizing, we want to make sure we maintain that. And there's a couple ways you can do that in AIDD. The first is to build a machine learning QSAR model of the activity, and I'll show you briefly how I did that. The other is to use a really powerful tool we have, a 3D ligand volumetric shape and pharmacophore similarity tool, just to make sure you're kind of filling that same binding pocket with the same pharmacophores in the same places. And the third is, if there's part of your series that some of the atoms are crucially important in that position, you can dictate that every molecule that AIDD designs has that specific substructure. All right, I think the best way to do this is just to hop right into a demo. So let me hop back here. I want to show you the data that I put together. Again, before this, I wasn't really all too familiar with small molecule GLP-1R agonists, but I went through the literature, and what I did is I think I pulled out a bunch of different papers, but I ended up using data from 4 or 5 studies. And this is important. This is where your biological insight is really important, and knowing the target well. So I only used data from papers that had the same activity assay. In this case, it was the same cell type expressing the same GLP-1 receptor and using the same cyclic AMP accumulation assay. And it's always better if you don't have to combine data from different labs. So again, if you have your own internal series and it's all measured in your hands, the model that you build will be even better. When you do have to combine data from different sources, it's nice to have an internal standard, right? In this case, that parental compound was tested by, I think, 4 of those labs, and in every case in that assay was within an order of magnitude. So you could scale, data one another. I didn't feel the need to scale in this case because the parental compound, that internal standard control, was so similar among them. So I felt comfortable combining all this EC50 data into one data set. The other thing I actually paid close attention to was I made sure that each of these had some sort of structural information about where they bind. I wanted to make sure they were binding in approximately the same binding pocket, right? You don't want to be trying to create a model for compounds that bind in 2 different places, because that model is going to fail. And so that's what I did. So I will show you the model that I ended up building, but I want to show you our tool, AdMed Modeler tool, to do this. In this case, we're thinking about building a model on the EC50 data. That's fine. It doesn't like the spaces. And so you bring up AdMed Modeler, and there's a bunch of different choices here. We have a pretty wide range, so it would make sense to logify this. And you can see all the different descriptors we have, even more than this in AP13, some really good, strong new descriptors coming out. And so it goes on a random seed, doesn't really matter. You can dictate how many processors you want to use. Its default is to one less than it recognizes on your computer, things like that. So we can do both regression and classification models. In this case, it recognizes that we don't have binary data, so we're not doing a classification, we're going to do a regression. We support multiple different types of machine learning algorithms. Almost all of our models are built with these artificial neural networks. They've worked great for years. In AP13, we have the ability to bring in models from XGBoost. If anybody's familiar with that, it's a newer machine learning tool for building models, and has some success in areas where the artificial neural networks sometimes fail. Your test set selection, always important. How you sample the data for training and test sets to make sure that your data, there's always the left out test set. And so you can use Cohonen mapping, you can set the minimum set test size, you could do stratified random sampling. There's a couple different things you can use. Also, for the best way to pick the descriptors, we have different mechanisms as well. For big data sets, truncated linear analysis goes really fast. For smaller data sets, genetic algorithm performs really well. And on the next slide, you can sort of go with the defaults. And what you're going to do is, I'll run it in a second, but you're going to build a bunch of ensembles of artificial neural networks here, starting with 1 neuron up to 4 neurons with different inputs, numbers of descriptors. And you can do these. We actually have a fancy updated version of called Linear Boost artificial neural networks that one of our modeling gurus invented himself. It works really well to help spread out the predictions toward the outer regions of the observed range. And again, we won't run this whole thing, but you can just start to see, why not? This runs really, really fast on a data set of only 50 some compounds. I'm sure these models aren't going to look that good, but you can see the different metrics we have, root mean squared error, MAE, Spearman rank, R squared, Q squared, different metrics you can have to look at it. You can always pop this open. Honestly, not a terrible model for not trying to finesse it at all. But you can see the kind of model you can get and the metrics on it, right? And so let me go back to this real quick. I'll bring this up. This will probably look better. So I built a QZAR model. I played around with a little bit with these, 50 compounds or so. And you can see pretty good performance. Again, this is about the bare minimum size, I would say, to try to build a QZAR model. We'd certainly like to have over 100 data points if possible, but it's a starting point, right? The other thing we can do, and I will show you this in the demo as well, but if you have a crystal structure where you have a co-crystal structure with a ligand, you can use, take that ligand out of the PDB and use that ligand to make sure that anything you design is in that same sort of volumetric space with the same pharmacophores. And I'll show you that as well. And the third example here, the structural filter, like you saw that this specific group was in all of those molecules in that chemical series. And I honestly don't know if it's essential or not, but for the sake of argument, let's say that we know that this group, that this substructure here is really vital to the activity of these compounds, and we don't want to lose it. And I'll show you what these other things mean here as we go to do it. All right. Let me hop back into this then. Let us go to this data. And the way AIDD starts is with a seed. So we could pick a bigger seed, but it's often nice to start with a smaller seed. All right, so this was a seed I wanted to build off of. And actually this is a AIDD run I already did, and we'll come back to it, but I want to show you everything that went into it. All right, so if you hit this AIDD button, this pop-up window comes up, and this is where you pick the parameters that you would like to optimize. And I've just started to populate this already. You can see a bunch of different things. We have a synthetic difficulty model. This is that 3D similarity I was talking about. I like to use our ADMET risk model because it brings into one score 22 really important other models that include, you know, like different permeabilities and solubilities and the main toxicity models, things like that. But really, almost all of our regression models are available here. This is that QSAR model that I built. This guy here. And what else do I have? Anything else I have in here? So the goal here, going back to the main goal here, was to keep the efficacy, the good PK, but also to enhance the blood-brain barrier penetration. And so a couple models that we can do that with. We could use our log BB. It's here, which is a pretty good model. AP13, we've improved it vastly. So if you were going to use it, wait till you download AP13. Much bigger data set built on, trained on, and I think better performance. However, like you saw in the first part of the talk, we also have the brain to plasma partition coefficient, the KP. And we can actually optimize based on that score. And so here we've clicked the tissue selectivity brain. So this is actually going to send all of the compounds we generate out to the HTPK calculation and bring us back that KP and use it to optimize. So we need to pick the parameters for the PK, the HTPK, whether we want this to happen in rat, human, or mouse. And again, in AP13, we could pick dog or monkey as well, and just the dose that we want to start at. So let's go human. I wish I remember what dose it said, but it wasn't that high, right? So let's just go with a 1 milligram dose. The other things we can do is we want to look at the 3D similarity. And so I pulled this compound out of one of those papers. It was related to the original discovery of that, the parental compound there. And I pulled it out of the PKD, the protein database, and turned it into an SDF file. And here you can see that compound. Again, you can hook ADMET Predictor up to any visualization software you want in the preferences setup. I just have it mine to Discovery Studio Visualizer. But here you can see what that compound looks like in its crystal bound pose. And so that's what we're going to try to match to. Let me go back over here. There's a couple different things. We're going to go with defaults here. But what I want to show you is the feature file, the pharmacophore feature file. This is our standard feature file that basically just looks at hydrogen bond acceptors, donors, positive, negative charges, and rings. And so not only are we going to match to the volumetric space, we're also going to try to make sure these features have a weight as well in there as we're designing these molecules. All right, so a couple other things we can do here. We have two different ways of multi-parameter optimization. The first is called a Pareto optimization, where you're looking in multiple dimensions and taking only those that are non-inferior in at least one dimension and adding those back into the next generation to optimize the molecules further. The other is a multi-criteria decision analysis tool. The Pareto weights all of your objectives the same. The multi-criteria decision analysis tool, MCDA, allows you to weight them differently. So 3D similarity, we want to maximize that. You can put a capping value on it, so the optimization algorithms don't get stuck in an energy well. So if we're going to maximize this one, 3D similarity, we don't need it to be doesn't matter if it's more than 0.9 out of 1. We want to keep this pretty high. Let's go with a 5. The at risk, we want to minimize. We'll put a capping value of 2, put a weight of 2. The tissue selectivity to the brain. Let's say we'll weight it as a 5. And this QSAR model, we'll also give it a 5. I don't know. Sorry. Yeah, okay. So then we can hit next. Here's where we can add the query filter, right? And so if I bring up MedChem Designer, you can see the seed pops up here. I chose the seed because it was the minimal of this substructure we decided was really important for activity, but we want to be able to build off of this. And we know that for most of the molecules, there is another ring that starts here, right? And so if I showed you back here, I turned this into, let me show you how I end up doing this. So in this molecule, what we can do here is right click on any atom or any bond, and there's a bunch of things we can do to dictate it. So for instance, we know that we want this to be part of a ring, right? And so if we put R2 here, that means it's going to be bound to 2 ring atoms. We're going to build a ring here in all of our molecules. But we also saw in our compounds that this could be a carbon or a nitrogen, so that we can go here and hit the table and say, all right, we will allow either a carbon or nitrogen. Okay. So what's going to happen now in the optimization scheme, we'll send this back over, is that we are going to evolve this from this seed primarily right here by starting to build a ring here and then expanding out. While this structure has to be in all of the molecules. Now, we could say that we want no changes at any of these positions. In fact, let me go back and I'll show you how we might do that. If we want to make sure we're not evolving, like here, for instance, we could say set substitution count as drawn. So no changes are going to happen there now. And we could do that with all of these if we wanted to make sure that we didn't evolve the molecule here. We won't do that just yet. I should point out that we have worked really hard on our drug-like filter, so any molecules that come up with really bad, groups that don't go together, hopefully will be filtered out here. You're always going to have to do some manual checking, but it shouldn't be that bad. And so here you can decide the number of optimization generations. I'd say a typical run for us is about 50 generations. You can change the number of molecules you make in each generation and things like that. Let's put this in just in a temporary somewhere. That's fine. We can just keep it here. That's fine. It will write out in the intermediate file, so you can actually watch as the molecules evolve over time and see how the algorithm's working. We'll just keep all of this as is for now. We'll just run 2 generations so you can start to see how fast this is, and then I'll show you the results from a 50 generation run. That's fine. And so what's happening here is that in the background, it is running this optimization and trying to create these molecules. And again, as I said, it starts with the seed and then it adds different transforms until it builds out and makes these scores better. And what you get is an output like this. Let me make these rows a little bit bigger. And so you can see in this specific run, I actually had not only that QSAR model, I should have mentioned the QSAR model that I built was actually on a negative log of the EC50. So a 9 would mean a 1 nanomolar EC50. A 6 would mean a 1 micromolar EC50. So you can see here, this is somewhere between 1 and 10 nanomolar. I did the at risk as an objective. I did the brain tissue selectivity as an objective, the 3D sim as an objective, as well as one I didn't do in that setup, also the oral bioavailability. Again, important, we want to the oral bioavailability, if it's predicted to be high, probably a lot of the other PK parameters are pretty good too. Obviously not the end all and be all, but one way that we can maintain the decent PK predictions, right? And so it spits out all of these, and then we can, hit the admit button and calculate all the other properties like we've seen before. But even then, how do you start to optimize this, right? How do you prioritize? So, for instance, we have a couple tools. Over here, you can use these sliders. We can instantly cut off things that have below 70% predicted oral availability, right? We can cut off things that are less than 10. We want to make sure it's less than 10 nanomolar potency. The admet risk, let's make it less than 5, right? So we can create these sliders. It shows you the range. You can create the sliders, and you can see quickly we've gone from about 500 compounds down to 58. So one quick way to optimize. Now, let's say we had some other information we wanted to bring in. Let's say we had like docking scores, or maybe you have your own retrosynthetic score. I should mention we have a new retrosynthetic score coming out in AP13 hopefully as well. But in this case, let's add this. We also have a synthetic difficulty score that's very close to the ERDL score, that the synthetic accessibility score, if you're familiar with that. So we can calculate that real quick. And so we have a synthetic difficulty score. But let's say you had docking scores from Schrodinger or something, and you can import those really easy. File, import compound attributes. So as long as you have the identifier the same, you can open a text file or an ST file, and it has that same identifier and it has a docking score. You can bring that in as a column. So any outside data you have, very easy to bring into this spreadsheet. And now, let's say we want to really pick out the best. And again, sort of a multiple parameter optimization. All right, so we're going to add a compound attribute to all spreadsheet rows. And you can see, like we did the synthetic difficulty, there's a bunch of different things you could do. You can create principal component analysis. The one I want to show you here is our multi-criteria decision analysis ranking tool. So this is going to pop up. Boy, this looks a lot like AIDD, right? But again, here is a way to weight the criteria you want and bring in different things. Like, again, like if you have the docking score, you can bring that in. Here, we're going to bring in our synthetic difficulty. Maybe if I can find it. There it is. We certainly want our admet risk here. We want to make sure admet risk is low. Let's do let's definitely do the GLP. It's easier sometimes just to go along like this. There's our GLP. And this time maybe we want to use the log, blood-brain barrier. And again, we can decide whether we want to maximize and add a weight to this. Synthetic difficulty, we want to minimize, we'll leave it at 1. At risk, we want to minimize. This, we want to maximize, and we'll keep a weight of 2 or 3 here. In the log BB, we'd like to maximize this, and let's add a weight of 3 as well. So honestly, very customizable for whatever project you have, whatever you're trying to do. You have a set of compounds that you need to prioritize. In this case, it came from our AIDD engine, and so they've already been somewhat optimized on many of these. But as I said, you could take hits from a high throughput screen and then run it through this MCDA tool. And so what you see here is taking each of those criteria into account with its specific weights. You get an MCDA ranking where the lower the ranking, the better. And so when we look at these, the combination of don't worry about the seed, it just ends up popping up there. the combination of those comes up with the best compound looks like this. I have no idea. I haven't really looked through it, but it is a way to customize your multi-parameter optimization. And again, you're going to have certain criteria for each project that are probably similar, but might change a little bit for each project. So I know a lot of big pharma try to come up with, you know, like a QED score for a compound, or you know, I just heard a talk the other day from Merck where they try to bring a dose number into the early drug discovery prioritization, and that just is basically an efficacy number combined with solubility. This gives you some real flexibility to pick the parameters you want to optimize for prioritization. So I think with that, I will finish the talk. You know, showed you a little bit about how to evaluate some leads with ADMET Predictor and HTPK, and how to optimize with AIDD, prioritize with MCDA. How about that for a lot of acronyms real quick? The next steps in what you'll hear about in future talks from this series is how to use full PBPK modeling to deliver the compound at the right dose, at the right concentration, in as few iterations as possible. How to use QSP modeling, and specifically our obesity sim model, to set up a clinical development plan to increase the likelihood of you know, of success of the compound, and specifically acquisition or out licensing, depending on you know, the stage of your company. And finally, some of the tools we have for adaptive learning for protocol and clinical trial optimization. If you're interested in any of this, please feel free to reach out to me. I love to talk to people about these tools, and you know, my background in academic drug design. It's something that I really love doing, and I've created a series of videos too. Marketing made me do on YouTube to answer some questions and some tricks and stuff that you can do in ADMET Predictor. So I'll stop there and take some questions. Thank you.
Fantastic.
A couple of folks hanging in there. I appreciate
Yep.
it.
A
You can
couple
see
dropped
I
off
tried
at
to
lunch.
edit it. Yeah, I tried to edit it here and there to make it shorter, and it still was that long.
It's okay. It's very good. It's actually something that people will come back to. So a couple questions are: we've got multiple camps of potential users of your scientific software tools. One would be a mature multinational biopharma. One might be a startup. Could you kind of just give the lay of the land, how maybe a multinational biopharma who's got all these tools, they've built their own, they've got this, how that can dovetail into what they're doing, and then maybe how the smaller company or organization can get maximum use out of your tool set, something like that.
Yeah, John, that's a fantastic question. And I if anybody's still listening out there, I didn't feed him that. So I'll give you a couple examples from like the very smallest use in big pharma. As I mentioned, our PKA model, you know, just sort of is the industry standard. We just won the Eurosample Challenge again. Many of companies will buy particular models that they really like, that perform well for them, and we have many ways to access the software and the models, one of which is through an API. And so they'll often use the API to get predictions from our models and build that directly into their internal framework for their decision making. So from as simple as that, you know, just picking the best models and building it into what you already have. We have a couple customers that will build these predictions in the other software packages like Schrodinger Live Design. They really like the you know, the ability to look at the molecules, but also have our ADMET predictions. To the far other end of the spectrum, you're a small startup. You know, you mentioned I was at Terray Therapeutics, which spent a lot of time and a lot of money, and still do, to really build up their AI and machine learning teams, right? Not every company has that expertise. Not every company has that budget for it. And what I see is we offer this off-the-shelf package that's very customizable to the needs of any startup biotech that has all of that AI machine learning built in for all the early stage drug discovery stuff that you need. So you can just take ADMET Predictor with AIDD and HTPK, start designing molecules, start optimizing, and have the strength of you know, a whole team of people behind your small startup.
Yeah, awesome. That's great. There's when you really get into the science and obesity, diabetes, type 1, type 2, types we don't really even know, haven't classified yet, but there is definitely a continuum of, let's say, disorders when it comes to diabetes. And I can just speak from this from personal family experience. What are you seeing from a correlation or an investigation, and how is Simulations Plus maybe preparing and trying to understand that environment more than it is today? And maybe you can't say, but I'm just kind of curious, given the power of your tools and where, when Scott put up that slide on the history of obesity research, it's come a long way, and it's taken a long time, and a shift in thinking and things like that. So I'm just curious what you're seeing or what you're thinking.
Yeah, it ask this question again when Scott does his webinar too, because he will give you a much more detailed, thoughtful answer than I will. As you saw with my history there, I'm more in oncology, and actually I did my postdoc in nuclear receptors. So
Right.
the one thing I will mention here is something that Scott did. How many different disorders are related to obesity, and the fact that what I find fascinating is the fact that we have effective treatments now, and we're able to, you don't ever want to say prove, but we're able to support those hypotheses that we've had for years, that all of these conditions will get better if we reduce obesity. One thing that I actually worked on a decent bit, I worked on androgen receptor and andrology. That's how I got to prostate cancer in oncology in the first place. But PCOS is a very androgen-dependent disease, and I think we're starting to see some the relationship there. Some better outcomes with the decrease in obesity.
Yeah.
That things
Okay.
that some of the things I'm fascinated by.
It wasn't an easy question to answer. It was just, but you
No.
gave some nuggets, so I really appreciate that. I think that there's a lot of people have to leave at the top of the hour. It's their lunchtime for whatever reason. I'm not sure if any questions have come in through the live part. Mike, any questions from there? No.
There was one question there about sort of how our models are dependent on charge states, and I answered
Yeah.
that briefly. But for everybody else, yeah, we actually have a pretty extensive list of ionization descriptors that influence a lot of our models. So
Yeah.
again, as I said, our pKa model is the best, and that influences a lot of these other models too. That helps the performance of our different models. And real quick, I'll say, one thing I always tell people who are trying out the software: if you have, your own internal chemistry series, try the software, load your chemicals, load your different, series, see how our models respond. If you have a whole bunch of those red flag ones, well, maybe that particular model isn't so great. However, we have a really nice system called the Delta model system that can help bring in and incorporate your local chemical space and integrate it with our global model. We've had a lot of success with that with clients as well.
Okay. Now I've worked in this space earlier in my career by hand, built the neural networks, did all of this back in the early 2000s. One of the reasons I went on an adjacent journey is because I didn't have enough model quality data, and bottom line, the model quality was missing and the quantity was missing.
John,
Given what
even today, data is still the limitation.
I know, and this is my next question for you. You mentioned 3 companies that said, hey, they gave us their pKa data, and and it made a huge difference, right? Why can't we get the world to get to put a program together to get more of that model quality data to help further along the discoveries for everybody and everything else? I mean, is this something that Simulations Plus could go after? Is this something where we could get a community together? What are your thoughts on that, Jeremy?
Yeah, that's another great question, John. So there has been huge efforts in the last, well, several years for sure, but before that as well, with federated learning, right? Trying to come
Right.
up with data sets that everybody can use. The Polaris group right now for ADMET or ADME data, in vitro ADME data is working hard on standardizing that. As I mentioned, I'm writing this data. I created this database of PK endpoints, and so we've reached out to there's a handful of other groups who have put together some PK databases as well. And we are actively working with them right now to try to create this big public data set that people can use. It's
Awesome.
difficult. It's
Yes, it
difficult
is.
because how to integrate data from different sources becomes very difficult. What information you need, how to present it. And one of the reasons our models still outperform a lot of other open source models is we take the time to go through and read every primary literature source and make
Right.
sure the units are right and make sure that the data, there's no big outliers and things like that. So manual curation, we have great AI tools that do a great first pass. Kemble is awesome at a first pass, but there's so much errant data in Kemble that every time you need to go look at the primary literature still.
Right.
So it's a time and effort thing that only, in my opinion, only manual curation and putting in that time actually solves.
Yeah. No, I totally agree, and it's definitely an issue. Let's hope that our tools are getting better and this will reduce over time. But let's hope anybody listening here has the wherewithal or even the ability in their organization to say, hey, we could contribute to this. We could make this a reality. So that's the great hope.
Yeah.
Awesome. Well, phenomenal job. This is exciting. Everything looks great for somebody who did this in his early days and got a lot of people excited. You guys have gone to the nth degree, which is phenomenal, fantastic to see.
John, I
And
just wanted to mention really quickly before in this, you were
yes,
talking about doing neural networks, decades ago. Our founder, Walt, actually started this company. It's nearly 30 years ago. He was
yes,
aerospace engineering and using neural networks to understand, airflow and things like that, and then decided that he could use it for predicting properties of small molecules. And so we've been using neural networks for nearly 3 decades to do this.
yes. It was exciting work then. We didn't have the internet in 99 and 2000, but we had Power Fives, we had Craze, we had, I was at Merck, and we were one of the few companies in the world really driving in silico first approaches. I worked with amazing people. Um, and like I said, and then I had these lessons learned, which by the way, unfortunately, people are still having them today. Like, we
We
were all in
relearn them
this at
all the
that
time.
point. Yeah, and they're repeating these, these problems and these mistakes. So I just went on this evangelism of creating, model quality data environments, fair, findable, accessible, interoperable, reusable, reproducible science, right? That's the ultimate goal here. We can do that. So, and then I went on and went to Schrodinger to build live design. So you mentioned that before. It was for me, it was a passion to enhance the usability or the experience that med and synthetic chemists have together, because so this is all, and now your models are in there, you guys, it's all part of that big team picture that it takes to discover new drugs and new
That's
therapies.
right.
So
Yeah.
it's phenomenal.
I love live design too. I guess I probably shouldn't say that, but it's lovely.
Yeah, it's all good. There's room for this. It's all
Yeah.
room here for
But
everyone.
as you said, this is about breaking down the silos, especially in the bigger companies where it's, very departmentalized. This is something that everybody can look at on the same team and work together.
Right. Well, Simulations Plus is a top tier, best of the best, company, and you guys are producing high quality products, and it's not like you ever let off the gas, and that's the exciting thing to see here. So really looking forward to the next webinars in this series, and honestly, I've proposed a roundtable discussion when we get through this, and I think it's going to be really exciting. So I'm looking forward to that.
Thank you very much for hosting, John.
All right. Go back to bed, Jeremy. Thank you
Yeah.
so much. And thanks
See you,
everybody
everyone.
for attending and your time. Take care.